Hello everyone, welcome to the Power Hour series, and today our topics are photosynthesis in higher plant, respiration in plant, plant growth and development, which is also known as PGD. And in today's session we are not covering the entire topic, but in photosynthesis we are going to cover one very important topic, that is a light reaction. So today in first half we are going to cover a light reaction. In second half, we are going to cover a glycolysis from respiration in plant and PGR, that is the plant growth regulator. You know that there are 5 types of plant growth regulator, and those 5 types of plant growth regulator we are going to cover in third subtopic. Now, without wasting any time, let's start with light reaction, or also known as photochemical phase of photosynthesis. Now, if I told you photosynthesis, directly in our mind there is a reaction came, and this reaction is 6 CO2 plus 12 H2O in the presence of sunlight and chlorophyll A, in the presence of sunlight and chlorophyll A gives us C6H12O6, that is a glucose molecule, plus 6O2 plus 6H2O. Now, is that this reaction is simple like I draw here? No. For this reaction we have to follow 2 reactions, that is a light reaction as well as dark reaction. So what is the location of light reaction and what is the location of dark reaction? Question is that what is exactly a location of light and dark reaction? If I am saying photosynthesis takes place in the plant, green part of plant, that is most probably leaves. In leaves there is a cell name as mesophyll cell, and in mesophyll cell there is one organelle which is responsible for photosynthesis, is known as chloroplast. So I am drawing here the structure of chloroplast. This is a double membrane structure, and this double membrane structure have 2 important parts. One is this stalk-like membrane is known as grana, and this part is known as stroma. So grana and stroma are the 2 parts present into the chloroplast, and the information for you is grana is responsible for light reaction and stroma is for a dark reaction. So in grana there is a reaction takes place, this reaction is known as light reaction. In stroma there is a dark reaction. Now what is exactly a light reaction and dark reaction is? I am customizing this slide, adding one blank slide and writing here the importance of light reaction and dark reaction. Here I am going to start with photosynthesis. And if I am talking about photosynthesis, there are how many reactions? 2 reactions. One reaction A is a dark reaction, and second one is B, light reaction. So here it comes in our mind that which reaction takes place first. This is not the thing that which reaction takes place first. Both reaction takes place simultaneously, but the product of light reaction is useful for dark reaction. What I am saying, the product of light reaction is useful in dark reaction. So what are the products of light reaction? First of all, let's discuss what are the products of light reaction. The products of light reaction are ATP, that is adenosine triphosphate energy source, and second product of light reaction is NADPH, or we can say that NADPH2. It is reducing power. Now why I called NADPH2 as a reducing power? Consider I am a NADPH2. Consider I am a NADPH2. So I have H2 molecule or hydrogen molecule with me. If I donate this molecule to any substance, if I am donating this molecule to the, consider a person name as X. I am NADPH2 and I am giving this H2 molecule to a person X, then what is going to happen with the person X? It get reduced because of acceptance of H2 molecule. So NADPH2 is a reducing power. So for photosynthesis, for dark reaction, we need ATP and NADPH2, and this ATP and NADPH2 takes place in a Reich reaction, means it is formed in the light reaction. Now I am writing here two forms of light reaction. One is named as cyclic photophosphorylation. One is cyclic photophosphorylation. Second one is non-cyclic photophosphorylation. One is cyclic photophosphorylation, second one is the non-cyclic photophosphorylation. We are going to cover one part, that is non-cyclic photophosphorylation first. Ultimately we can understood the cyclic photophosphorylation from the non-cyclic photophosphorylation. That's why we are going to cover a non-cyclic photophosphorylation right now. And what is going to produce after non-cyclic photophosphorylation? The two molecules, one molecule is ATP and another molecule is NADPH2. And how exactly it carried out, let's see in detail. Now I am making this screen little bit suitable for a non-cyclic photophosphorylation. Non-cyclic photophosphorylation, first point, it involves both photosystem 1 and photosystem 2. What I am saying, non-cyclic photophosphorylation involve photosystem 1 as well as photosystem 2. So if I am talking about second point, PS1, it means it work on a 700 nanometer wavelength sunlight. Now if I am talking about PS2, then it works on 680 nanometer wavelength sunlight. Now PS1, PS2, probably you understood about the PS1 and PS2. Now second point, third point, non-cyclic photophosphorylation. This non-cyclic photophosphorylation responsible for production of one molecule of ATP in one cycle and one molecule of NADPH2, or we can say that NADPH. Next important point, I am writing here that non-cyclic photophosphorylation, non-cyclic photophosphorylation required water, means a splitting of water carried out in case of non-cyclic photophosphorylation. Now here is a blank space. I am drawing here a non-cyclic photophosphorylation. Okay, I am drawing here a non-cyclic photophosphorylation. Just focus on a board. Okay, I am writing here a non-cyclic photophosphorylation. So I told you before also that it involves both photosystem 1 and photosystem 2. Consider it start from photosystem 2. How much nanometer light is required for PS2? There is 6. For example, this is sun. 680 nanometer of sunlight. Now, consider this as a reaction center. PS2, a reaction center molecule of PS2 is chlorophyll A. Reaction center is chlorophyll A. Now, other molecule is also present here. Those are the accessory pigment like chlorophyll B, chlorophyll C, chlorophyll D, bacteriochlorophyll, bacterioviridin. The function of this what we can say accessory pigment is just to absorb light and pass to the chlorophyll A. The main molecule, or we can say that reaction center of non-cyclic photophosphorylation PS2 is chlorophyll A. Just understood that point. So 680 nanometer of sunlight is absorbed by all of the accessory pigment. It is transferred to the PS2 chlorophyll A molecule, and chlorophyll A molecule have one electron in an orbit. One electron in an orbit. Because of this much of energy, sunlight, the electron get excited. This electron get excited, and this electron is accepted by primary electron acceptor, and the primary electron acceptor for PS2 is phosphorylated. Or in some book it is written as, coenzyme quinone, or some in book it is written as coenzyme quinone. But according to NCERT, or according to what we can say a concept of non-cyclic photophosphorylation, just remember the term phosphorylated. If the question is asked into the NEET examination, that is which is first primary electron acceptor in case of PS2, then your answer should be phosphorylated. Now 2 electron is accepted by phosphorylated. This phosphorylated is transfer this electron to the PQ. Now, what is the long form of PQ? That is the plastoquinone. Plastoquinone transfer this 2 electron to cytochrome B6. Cytochrome B6 transfers this electron to cytochrome F. And in between this transfer, this 2 electron, in between cytochrome F and cytochrome B6, there is ADP molecule fused with inorganic phosphate and form ATP. One molecule of ATP formed during this reaction, cytochrome B6 to cytochrome F. Now, what is happen next? Cytochrome F giving this electron to the PC, that is the plastocyanin, and plastocyanin transfer this electron to the PS1, and you know that PS1 absorb light, which is how much light? 700 nanometer light. Also PS2 have some accessory pigment like chlorophyll B, chlorophyll C, bacteriochlorophyll, bacterioviridin, and all of the accessory. Now, this 2 electron which is accepted by PS1, 2 electron due to the absorption of 700 nanometer light, it is also excited and it is also absorbed by a primary electron acceptor. But now the primary electron acceptor of PS1, is FRS, and FRS stand for ferredoxin reducing substance. 2 electron is absorbed by ferredoxin reducing substance. Thus electron transferred to the FD is the ferredoxin, and given to the NADP molecule. So because of 2 electron, it becomes NADP di-negative ion. I hope so you are getting my point. Everyone is joined in the meeting. I'm hopeful that you are getting my point, that NADP convert into the NADP di-negative ion. Now, electron is with NADP molecule. NADP becomes a NADP di-negative. Now, reaction is over here, but this PS2 didn't have electron with it. So, it required another electron for next reaction, next non-cyclic photophosphorylation. So here come to the last point of this non-cyclic photophosphorylation, that it required water. It required water. Now H2O molecule. Now here is the role of H2O. H2O molecule is split. It is a photolysis of water, which is called as photolysis of H2O. And H2O is split into 2 electron negative plus 2 proton positive plus half O2 molecule. Now, this 2 electron required to PS2. So I am giving it to PS2. Here is moving towards the PS2. Okay. So there is a requirement of electron fulfilled. Now this oxygen comes out from what we can say, this is happen in a grana. So out of grana means it comes into the stroma, ultimately stroma to cell, cell to the stomata, stomata outside the plant body. Like that this oxygen move. But what is going to happen with the 2 proton molecule or 2 H plus molecule? The complete reaction is in front of you. What is exactly a function of those 2 H plus molecule? Those 2 H plus molecule, those are the positive charge. And which is the negative charge molecule we have here? NADP di-negative. So directly those 2 molecule of H plus going to NADP di-negative, and it becomes now NADPH2. This is what exactly a reaction is. 1 ADP molecule, 1 ADPH2 molecule. I am writed here in involve. It involve both photosystem 1 and photosystem 2, responsible for formation of 1 ATP as well as 1 NADPH2 molecule. This is a complete reaction of non-cyclic photophosphorylation. I hope so you are getting my points. Yes or no? Yes. If you are getting my point, write in comment section, yes sir, I am getting your point. Give me a 2 minute of time. Just note it down this. This is a very important reaction. Give me a 1 minute of time. I am just taking a little water and let's continue. So here Dilip is saying, sir, please put 10 years of PIQ for bio. Yes, definitely. Dilip, it is possible. We can definitely we can do this for you. Games Mind saying, yes sir, 680 nanometer. Ashish saying 680 nanometer. Correct. Okay guys, let's move to the non-cyclic photophosphorylation to the cyclic photophosphorylation. So I am saying that if I am talking about the cyclic photophosphorylation, just I need that previous slide only. This slide we have to remove, and this slide we are going to copy here. So I'm going to erase this much of portion. And writing here about cyclic photophosphorylation, first point, it involves only PS1, only PS1. It works on 700 nanometer of wavelength. 700 nanometer of wavelength. Third, only 2 molecules of ATP is produced here. No NADPH2 production. Reducing power is not produced during the during the cyclic photophosphorylation. Only ATP molecule is produced. Now, 1, 2, 3, fourth point, very, very important point I am writing here, that it does not involve splitting of water. Now look, this is cyclic. Cyclic means the electron excited start with the journey and comes at same position, right? If electron is traveling in a loop, in a circle, in a cyclic manner, there is no requirement of splitting of water, because previously in that diagram, in that PS1, PS2 system, that is the non-cyclic photophosphorylation, the excited electron is not came back to the PS2. That's why we split the water, which is known as the photolysis of water. Here is the term. And due to this, we produced thus 2 proton molecule which is attached to the NADP dinegative, and NADPH2 formation takes place. But in case of cyclic, it is not going to happen. There is no photolysis of water. Look, this is the simple differences between two topics. We have to understand the small thing. And NEET is not, I am saying, a difficult exam in terms of knowledge and all, because look, it is easy. Not easy, we can say that moderate. 650 marks, getting 600 plus marks or 650 marks in a NEET is not difficult, because 140 MCQ out of 180 are easy to moderate. Just there is a high level of competition into the NEET examination. We have to do, or we have to put our extra efforts for the NEET examination. So believe me, understood the thing, success is never owned, it is rented, and rent is due every day. So do work consistently. Daily to daily practice is required for getting good score in NEET examination. If you understood my point, let's move to the next topic, which is cyclic photophosphorylation. I'm starting cyclic photophosphorylation with PS1 because it involves only PS1. If I am asking you a wavelength of absorption of PS1 is 700 nanometer, right? 700 nanometer absorbed by PS1. Here is molecule, accessory molecule present in the PS1. That is chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E, bacteriochlorophyll, bacterioviridin. Here is chlorophyll A, reaction center. Two electron moving in the orbit of the chlorophyll A. Due to the absorption of 700 nanometer, electron get excited because of high energy, and it is excited, and you know that which is the primary electron acceptor in case of P as 1. It is FRS, and FRS stand for ferridoxine reducing substance. Ferridoxine reducing substance pass this to the FD. You know that. Now there is a reverse process. This FD is not going to pass this electron to NADP. It is passing this electron to the PQ. PQ is a plastoquinone. PQ, and in between this reaction, because electron is passed to electron negative, to electron negative, to electron negative. In between FD and PQ, there is synthesis of ADP plus IP, giving us the ATP. One molecule of ATP is formed here. Now further reaction, plastoquinone pass this 2 electron to the cytochrome B6, cytochrome B6 to the cytochrome F. In between, because of this passing of electron, 2 electron negative, 2 electron negative, 2 electron negative. Here is a production of ADP plus IP, giving us a ATP. Second molecule of ATP is formed. Now the electron is passed from cytochrome F to PC, plastocyanine, and plastocyanine to PS1. The cycle is completed, guys. The electron started from PS1, ended up in a PS1. 2 electron negative. Thus 2 electron negative molecule comes to the PS1 in a cyclic manner. So only formation of 2 ATP takes place. There is no formation of NADPH2, and there is no photolysis of water in case of cyclic photophosphorylation. And now you understood the thing that due to the cyclic photophosphorylation, because of cyclic and non-cyclic photophosphorylation, PPR, what is happen? Formation of ATP and NADPH2 takes place, and due to the formation of ATP, NADPH2, thus ATP and NADPH2 used up in dark reaction, and this NADPH2 and ATP is going to produce C6H12O6 in dark reaction. Now my question is to you, my question is to everyone: how many molecule of ATP and NADPH2 required for formation of 1 glucose? Simple question. My question is very simple. I am asking you how many Pheophytin, primary electron accelerator. It is right. Games mind. Mayank is saying yes sir. Ashik Muhammad is saying, sir, I want just qualify NEET. Please tell me the important chapter in the biology. Important chapter is, we can say that genetics is important. Molecular basis of inheritance and principle of inheritance and variation. It counts nearly 6 means 12 MCQ, 48 marks from this, from botany section. Now, in case of zoology, biotech 1, biotech 2, 8 to 10 MCQ, nearly 40 marks. In case of 11th NEET, photosynthesis, respiration, which is we are covering here, anatomy, morphology, nearly 12 MCQ, 12 to 13 MCQ, so it count 50 plus marks. And ecosystem means ecology. Ecosystem, the I know that those topics are easy but high scoring. In NEET, there are 8 to 10 questions asking from this ecology section. So from ecosystem, biodiversity conservation, and organism population, probably 8 to 10 questions, so 40 marks. So 40, 40, 80, 80, and 40, 40 means nearly 130 to 140 marks from those important chapter. Other than that, for 40 marks, you have to cover other syllabus, right? Total, how many chapters are there? Total 33 chapter from 11th and 12th. So we have to focus on those 10 to 11 chapter from which 140, 150 marks is going to cover. Rather than this, if you are only want to qualify the exam, for scoring 170 marks, each and every topic, each and every line of NCERT is important. I hope so you are getting my point, guys. My question is very simple. Question is, how many molecules of ATP and NADPH2 required for formation of 1 glucose? So answer for this question is 18 molecule of ATP and 12 molecule of NADPH2 required for formation of 1 glucose. So I'm expecting this answer from you in chat section. I'm expecting this answer from you in the chat section, but let's move to the another topic. Now we understood about the non-cyclic photophosphorylation. It is also known as Z scheme. Understood the point? It is also known as Z scheme. Other than that, we cover all of such things, that splitting of water, photolysis, cyclic, non-cyclic, and all the things. Now, chemiosmotic hypothesis. Chemiosmotic hypothesis. What is osmotic? Osmosis is any molecule travel from higher concentration to lower concentration. Chemio, that is a chemical reaction. Hypothesis, that is the study. Now, this entire reaction of non-cyclic photophosphorylation, this entire reaction of non-cyclic photophosphorylation, it is carried out in grana. A stalk of grana is known as the thylakoid. It is carried out in the thylakoid. So in chemiosmotic hypothesis, we understood that this is outer surface of thylakoid, this is the inner surface. On inner surface, there is a H2O splitting complex. And there is a PS2 from which electron is transferred to the PS1. And due to the transfusion of electron, we told before in non-cyclic photophosphorylation that 1 ATP molecule is produced, but it is not produced directly. Just because of this electron transport, 1 proton molecule comes inside the lumen of thylakoid, and due to continuous process, a large amount of proton is present, into the lumen. Due to water splitting also, because water is split into 2 electron negative, 2 proton positive, half O2. So due to that also, proton is present into the lumen. So high concentration of proton in lumen. Outside there is a low concentration. With the help of high energy, this proton is travel from high concentration and low concentration, and through F0 F1 particle. You know that F1 particle have ATPase enzyme, and this ATP is responsible for formation of ATP. So we told directly here ATP is formed in between cytochrome B6 to cytochrome F, but exactly how ATP is formed is explained in chemiosmotic hypothesis. Then what happened with these two proton molecules which come with the high energy? This proton molecule binds with the NADP dinegative and form NADPH2. This is chemiosmotic hypothesis only. Now, I hope so first half is done. We are moving towards the second half. That is a glycolysis. What do you understand about the glycolysis? Here I am writing the word glycolysis. So, glyco is glucose, lysis is breakdown. Now, the breakdown of glucose into two molecules of pyruvic acid is known as glycolysis, and it is an anaerobic process. Anaerobic means it is not required oxygen. Now understood the thing. If it is an anaerobic process, it can be carried out in human also, means eukaryote, and it can be carried out into prokaryote also, that is a bacteria. So it is a common pathway. What I am saying, glycolysis is a common pathway for eukaryote as well as prokaryote, because it does not require any oxygen molecule. It means glycolysis is an aerobic process. So if I am asking you a respiration, in broadly if I am asking you the respiration, so what is the reaction of respiration? Broadly, if I ask you about the respiration, the answer is coming from class like, sir, O2, CO2, inhalation of oxygen, exhalation of CO2 is respiration. No, it is not respiration, it is breathing. It is breathing. Now, this oxygen we inhale going towards my lungs, and it is entering into the lungs, and carbon dioxide coming out from lungs. This is the external respiration. This is the external respiration. Now, the oxygen transfer from lungs to blood capillaries, means alveoli to blood capillaries, and blood capillaries giving a CO2. This is the internal respiration. Now this blood is flowing through the body, giving the oxygen to the cell and taking CO2 from cell. This is known as cellular respiration. So what is the respiration? Breathing, external respiration plus internal respiration plus cellular respiration combined known as respiration. Now in this topic, which respiration we are studying? Cellular respiration. So I am writing second title in the respiration, that is cellular respiration. That is cellular respiration. Now what is exactly cellular respiration is? What is exactly cellular respiration is? Understood the thing. Reaction I am writing here, 6 molecule of CO2 plus 6 molecule of H2O gives us C6H12O6 plus 6O2 plus, not plus. If you are writing this reaction, it is known as photosynthesis. Just we studied about that. Now this is a product of photosynthesis, right? This product of photosynthesis which is going to break, the product of photosynthesis is going to break. It means C6H12O6 in the presence of 6 oxygen molecule gives us 6CO2 plus 6H2O plus 38 molecule of ATP. Now what is ATP? ATP is adenosine triphosphate energy. This is the reaction of respiration, which we call as cellular respiration. So in cellular respiration, I am moving it above. In cellular respiration, we have to follow some steps. We have to follow some steps. First step is glycolysis. That is a breakdown of glucose. This is our topic, today's topic, glycolysis. Second thing is, we can say that oxidative decarboxylation, which is also known as link reaction. Then third one is the TCA, tricarboxylic acid cycle. Then ETC, electron transport chain or electron transport system, ETS also we can mention, or we can say that plus oxidative phosphorylation, complex number 5, oxidative phosphorylation. So after that only we get 38 molecule of ATP. In glycolysis, just glucose molecule is going to split into 2 molecule of pyruvic acid. 2 molecule of pyruvic acid. Now, this is I am talking about aerobic respiration. What about the anaerobic respiration? What about the anaerobic respiration? If I am saying anaerobic respiration, definitely it involves glycolysis. Yes, it involves glycolysis, but here C6H12O6 molecule is breaking down in the absence of oxygen and gives you 2 molecule of C2H5OH plus 2 molecule of CO2 plus 2 ATP. This is known as alcoholic fermentation. Alcohol is formed. Second, C6H12O6 in the absence of oxygen gives you 2 lactic acid molecule plus 2 molecule of ATP. Then this is known as lactic acid fermentation. So alcoholic fermentation and lactic acid fermentation. If I am talking about the alcoholic fermentation, if I am talking about the alcoholic fermentation, there is the production of CO2. And if I am talking about the lactic acid fermentation, there is no production of CO2. Can you tell me why so there is no formation of CO2 in case of lactic acid fermentation? Here, first one is the alcoholic fermentation. Second one is the lactic acid fermentation. No production of CO2 and production of CO2 in alcoholic fermentation. Anyone? Can you give me the reason why there is no formation of? Yes, I am waiting in the chat section. Sandro, NEET 2027, sir. Definitely this lecture will help you for NEET 2027 also, because I am covering a basic topic of glycolysis. Okay, so in class 11th, what topics are important for you: photosynthesis, respiration, morphology and anatomy. So in respiration in plant, glycolysis is very important concept. Stay tuned, Sandro. My question is, 2 molecule of CO2 released in alcoholic fermentation. Why not in lactic acid fermentation? Anyone in the chat section? Try to give the answer. No one know about that? Dilip, Game Mind, Ashi, Mayank, Ashik, Sandro. Anyone? Question is so simple. There is a release of 2 CO2 molecule in alcoholic fermentation. Why not in lactic acid fermentation? Who is going to try? No one. Okay. I am giving you an answer because guys, C2H5OH, it is a 2 carbon molecule, and 2 molecule of 2 carbon is a 4 carbon. Glucose is a 6 carbon molecule. Glucose is 6 carbon molecule. And ethanol is a 2 carbon, 2 molecule of 2 carbon means 4 carbon. So 2 molecule of carbon is going to be released in case of alcoholic fermentation. Now here glucose is 6 carbon molecule, lactic acid is 3 carbon molecule, so 3 carbon molecule, 2 molecule of lactic acid is 6 carbon. So how can we release the CO2 in the lactic acid fermentation? There is no possibility because lactic acid is 3 carbon molecule. This ethyl alcohol or ethanol is 2 carbon molecule. Understood the thing? Yes or no? Just write in the chat section. Do you getting my points? Okay. Now, what is exactly glycolysis? Glycolysis is a conversion of glucose into 2 molecule of pyruvic acid. And this is a complete reaction of glycolysis. Due to the short time, I am not writing the glycolysis reaction on a blackboard. We have this slide in which first glucose is converted into glucose 6-phosphate. Now, if I am saying glucose is converted into the glucose 6-phosphate, it means we required 1 phosphate for joining to the position number 6 carbon. So ATP required for this process, ATP convert into ADP. ATP convert into ADP, and 1 inorganic phosphate joined with the glucose to form a glucose 6-phosphate. Now, this glucose 6-phosphate is converted into the fructose 6-phosphate. There is a requirement of enzyme here. First hexokinase, second is the isomerase. Second enzyme is isomerase. Then fructose 6-phosphate is converted into the fructose 1,6-biphosphate. Here is also requirement of ATP. So we are saying that we need 38 ATP or 2 ATP in case of anaerobic. In case of aerobic, 38 ATP. And we are spending here ATPs. We are not getting ATPs, we are spending ATP. Why? It is called as investment, guys. It is called as investment. If you want to start a business in future, you qualify a NEET 2025 or 26 or 27, and you become a doctor, and you don't want to do a job 9 to 6 journey. You don't want to do this thing. You want to start your own hospital. So is it possible from day one you are getting the income? No. You have to build a building, taking equipment required for your hospital, hiring of staff, set up a medicinal facility. Thus all things required investment. But after 1 year or 2 year, when your hospital get established, then real income start coming in your hand, right? So first investment is required. Like the same way, for formation of 38 ATP, we required little investment. This little investment of spending of ATP. We are spending 2 ATPs here. Now fructose is converted into the triose phosphate. One triose phosphate is PGAL. Pagal. Okay? PGAL. And second triose molecule is DHAP. Now this is known as DHAP. Okay? DHAP is dihydroxyacetone phosphate. PGL is phosphoglyceraldehyde. Both are 3 carbon molecule. So those are isomers of each other. The breakdown required aldolase enzyme, and this is the isomers of each other. Now just keep DHAP aside and start with the PGL. This PGL convert into the 2 molecule of 1,3-biphosphate glyceric acid, and it required, or it responsible for formation of 2 molecule of NADP. Sorry, 2 molecule of NADH. Now here is a NADH molecule is formed, or NADH2 molecule is formed, and one NADH2 molecule gives you a 3 ATP. It means now ATP is coming. This 1,3-biphosphate glyceric acid convert into the 3-phosphoglyceric acid, and it produce ATP. Another ATP came. This 3-phosphoglyceric acid converted to 2-phosphoglycerate. There is no ATP. There is no formation of ATP because only 3-phosphoglyceraldehyde to 2-phosphoglyceraldehyde, mutase enzyme. 2-phosphoglycerate convert into the 2-phosphoenol pyruvate. There is no formation of energy, but 2-phosphoenol pyruvate convert into 2-pyruvic acid, release the ATP. So how much ATP is formed? 1 and 2 ATP. How much ATP is spent? 1 and 2 ATP. 2 minus 2, 0. But 1 molecule of NADH2 gives you 3 ATP. So T ATP plus. Now this is the one reaction. What we keep aside? DHAP. DHAP can convert into the PGL. This reaction takes place second time. And there is a 2 molecule and 2 molecule, 4 molecule of ATP. And 2 molecule of NADH2 means we can say that ATP spent 2, ATP formed 4, NADH2 form 2, ATP form NADPH2, 2 into 3, 6, 2 ATP spent, 4 and 6 means 10 formed, minus 2 spent is equal to 8. So if I am asking you a gross gain of ATP, it is 8. If I am asking about net gain of ATP, only 2. If I am asking about gross gain, it is 8. If I am asking about the net gain, it is only 2 molecules. You are getting my point? Guys, you are getting my point? Yes or no? I hope so you are getting. I hope so you are getting. Okay. This is all about glycolysis. Let's discuss last topic. Last 6 minutes we have. And the topic is PGR, plant growth and regulator. 5 types of PGR. 3 are promoter, 2 are inhibitor. Which 3 are promoter? One is auxin, second one is the cytokinin, third one is the gibberellic acid are the promoter. Which 2 are inhibitor? One is the ethylene, second one is the abscisic acid are the inhibitor. Why so? Because of their function. Auxin, forget the historical background. Let's start with the function. Auxin is responsible for initiate rooting in the stem cutting of the plant propagation. It is promoting. This is very, very important and crucial. 3 times asked. 3 times PYQ 3 times. Which hormone or plant growth regulator is promoting flowering in a pineapple? This is direct question in the NEET examination. Your answer is pineapple. To prevent fruit and leaf drop at early stage, good work. Promote the abscission. Abscission means fall of leaves of older leaves and fruit. Induce parthenocarpy. Parthenocarpy is formation of fruit without fertilization. In case of tomato, auxin responsible. They are used as a herbicide 2,4-D. It's a herbicide which is responsible for kill. What we can say, it kill dicot weed and it does not affect the monocot plant. If I'm doing farming and there is a production of wheat, I'm taking from my farm and there is dicot weed present on both sides of this plantation. If I am spraying 2,4-D, then definitely it will help to kill dicot weed and protect the monocot plant, that is a wheat plant. But we can't use this in field of dicot plant. Like if I'm doing the farming of what we can say a chana or tuar, understood? The dicot or groundnut dicot, and I'm spraying this weed, or I'm spraying this weedicide, or we can say that 2,4-D. There is a damage to the dicot weed as well as to plant also. So in monocot plant farming we can do this. Xylem differentiation also function of auxin. What is exactly apical dominance? They are saying in higher plant the growing apical bud inhibit the growth of lateral bud. Now, for example, consider this as a plant, this pane as a plant. This tape produce auxin, and due to this auxin there is a growth of height of this particular plant. Now, due to this moment the lateral bud formation is not carried out, and because lateral bud is not formed, there is a less production to the farmer. So what is the overcome means remedy action for this? We can cut this shoot part. If we cut the shoot, there is a stop growth of this plant and lateral bud or lateral branches start propagating and production is become high in the farming. This is known as apical domination. To prevent the apical domination, we do the decapitation, also known as the hedge making. Decapitation. Now second is gibberellin. What are the function of gibberellin? They cause increase in the length of axis. So they are used to increase the length of grape stalks. So increase the length of grape stalks. Look at the increase in the stalk. This is before, this is after. It is like an advertisement, right? Before I am looking like that. This is so sad, right? Here is before, after. Look at the size of apple, guys. This is a small apple. This is a large apple due to a very known plant growth regulator that is known as the gibberellin. To elongate and improve the shape of fruit such as apple. Yes, definitely it improves the shape of apple. They delay the senescence, so the fruit can be left on the tree to extend the market price. To extend market price is required, okay? GA3, this is an important point. GA3 is used to speed up the malting process in the brewing industry. Brewing industry, alcoholic industry, okay? Brewing industry. Beer, wine, whiskey, rum, brewing industry. GA3 improve the malting process. Malting process generally takes place in wine industry. So if you are thinking that, sir, you have a lot of knowledge of this beer, wine, whiskey. Guys, I am a BTech, MTech food technologies. So there is subject alcoholic beverages in 4th semester. So I don't have a practical experience, but theoretical experience I have. I hope so you are getting my point. The increase the length of sugarcane also, there is a requirement of gibberellin. Also spraying on the juvenile conifers, it can be helped. And all of this thing. I covered 2 basic hormones, cytokinin ethylene and gibberellic acid, in which cytokinin is a promoter, and ethylene and abscisic acid are the inhibitor. Just remember this thing. I hope so you are getting all the points from today's session. I tried my best to deliver you a content, a short and useful information of this 3 chapter. All the best for your NEET examination. Thank you so much. Take care. Bye bye guys.